Assalamu alaikum everyone. I'm Anika Jahin, a postgraduate student of Department of Biomedical Engineering at BUET. Today, I will discuss on the ongoing thesis. The topic is Angiopep-Functionalized Polymeric Nanoparticles for Enhanced Glioblastoma Therapy. Glioblastoma is an aggressive brain tumor, but we have a conventional chemotherapy technique available. In the chemotherapy, the temozolomide drug has been used, but there are mainly two problems. The first one is low blood-brain barrier permeability and systemic toxicity. So my thesis actually focuses on how we can decrease the limitations and increase the targeted drug delivery technique, also the drug efficacy. At first, Angiopep, a peptide, has been created here. So this peptide targets the low-density lipoprotein receptor-related protein on blood-brain barrier endothelial cells. So our methodology goes like this. First, nanoparticle design and encapsulation, then in silico physicochemical characterization, drug release simulation at pH 7.4 and 6, blood-brain barrier permeability simulation, molecular dynamics simulation, extended level prediction on heat map, and then preclinical studies and clinical studies. So in the results section, the first picture is about Angiopep encapsulating the TMZ drug. The second picture is zeta potential, and we can see a higher negative potential of the Angiopep nanoparticle, which means that it can easily go through the blood and then it can penetrate blood-brain barrier. The third picture is loading and encapsulation efficacy. So also the Angiopep nanoparticle has a higher loading efficacy. We can see here. The fourth and sixth picture is about molecular dynamics simulation. And in the fourth picture, we can see where the RMSD, root mean square deviation, is lower. That means our protein ligand complex has higher stability. So which is very important in this research. In the fifth graph, we can see cytotoxicity level. The green line is actually represent our Angiopep nanoparticle. So when we are using Angiopep nanoparticle, the toxicity is very lower. We can see here. And in the seventh graph, the green bar shows Angiopep nanoparticle. So in the tumor section, it is higher. That means when we are administrating the drug, it is actually available and it is biodistributed, and the distribution is like that. The tumor cells has been affected by the drug, but not other healthy cells. That means if we are using a very little amount of drug, which is encapsulated by Angiopep, it can easily destroy the tumor and not affect the other healthy cells. And the eighth picture is cross correlation matrix, which also emphasize on the molecular stability. And the ninth picture is about brain penetration heat map. So in conclusion, we can say that from computational biological method, we have seen the Angiopep-Functionalized polymeric nanoparticle has a higher efficacy on targeted drug delivery technique in the glioblastoma therapy. And for further validation, we have to make some preclinical trials and clinical trials. Thank you so much.